So we've spent some time talking about cell structures. We spent some time talking about the factors that microbes require to grow. Putting it all together, we're going to talk this module all about how do we control microbial growth. Straight up, it's not going to be the most exciting module, right? Of course, all modules are actually sort of exciting, right? But we're going to talk a lot about definitions and things like that, right? I always find stuff like this is the best really done hands-on, like in the lab, right? But we're going to get through this as best we can, right? So be prepared for some definitions, but try to see the application of this into your everyday lives, because the things we're going to talk about are definitely things that you're going to see all the time. Okay, so with that said, let's get started. So I think most of us have been told that microbes are everywhere. And of course, the truth of the matter is, it's true. Microbes are pretty much everywhere. And by everywhere, really what we mean is everywhere there's some sort of human contact, because of the fact that, it's going to be, they don't just appear, as we've learned, they have to come from something else, right? So it's typically humans or animals or some sort of mechanism that gets them there. And this is a neat little survey of where are the microbes that are in your car, right? And let's look at some of this stuff. Let's apply. So CFUs, you guys remember what that means? Colony forming units, right? So technically it's cells, right? If that's the easiest way to think about it, a CFU is a cell, but it's not really a cell, right? It's sort of a cell. And then so what they did here was they took a 6.5 centimeter by 6.5 centimeter area, or for those of us who speak American, that's 2 inch by 2 inch. Took a little cotton swab, and then they swabbed this area like that, and then placed that onto an agar plate, and then they counted the colonies, right? Colony forming units. And then the more CFUs we see on a plate, the more bacteria are present, right? And then if you kind of look at the pattern of this, right, we of course see stuff everywhere, right? But the ones that are most populated are areas that are going to get the most human contact, right? So stuff like your steering wheel, your center console, people are touching these things all the time, right? So those, of course, are going to have the most microbes. Now, keep this in mind. One of the things that I want us to realize is that, remember, we already learned this. Most microbes are not harmful, right? In fact, less than 1% are pathogenic. Less than 1% are pathogenic, right? However, pathogens are important to us, right? They do cause some great issues, right? We don't really want to get sick, right? So the idea behind this is that even though these microbes are not pathogenic, right, the more bacteria we find indicates really the more amount of human contact it receives, and the more human contact it receives, the greater the probability that there's going to be a pathogen present, right? So we call these types of surfaces fomites, which are inanimate objects that can transmit disease. Now, the key for a fomite is that it's got to be an inanimate object, right? So it's not just any object. Oops, what happened there? It's not just any object, right? It's some sort of inanimate object that transmits disease, right? So, and then again, the idea is that it comes from sort of some sort of human contact, right? Now, what are some examples of fomites we see in our everyday lives? Well, don't say center console or door latches or things like that, right? So just outside of the car, what are some of the fomites that we may encounter? Many of us are holding one right now, like a pen. We're on a table. Those would be fomites because they're these inanimate surfaces. You are touching them. And if you happen to be sick, you're going to put your pathogens on here, right? Whether they're bacteria or viruses. And then if you share this pen or someone else comes and picks it up, bang, they have now transmitted or received the pathogen, which is part of the chain of transmission. And we'll learn more about disease transmission later this semester, right? This chapter focuses all about the nature of control. How do we control the growth of pathogens? How do we know that we can safely use someone else's fomite? Now, I do also want us to understand this. For the most part, we are not going to be transmitting disease. If we're not sick ourselves, if we're not doing anything, these microbes that we see here, they're normal human skin bacteria, right? So they're not necessarily pathogenic, right? But, let's say we always have this risk factor, right? So if I see, there's 506 CFUs on the center console in my car, would I be freaking out? No. Do you know why? They're probably my microbes. I put them there, right? If I didn't do it, my family members did. We all have intimate contact with each other anyways, so it's really not a big deal, right? But let's say now you're in the midst of this global pandemic, and then you lent your car to somebody, right? Or it got serviced somewhere, right? And maybe at that point, then there might be this introduction of foreign microbes. Maybe then we would consider doing things like disinfecting or sterilizing. And we'll talk about the difference between these words a little bit later, right? So there's always these certain risk factors that you have to place on things. In general, don't freak out about stuff, right? But certain surfaces are definitely going to warrant a little bit more caution than others. Okay, when we talk about microbes, we assign them a biosafety level, right? And this is basically this risk factor, right? Sometimes we'll say, oh, this microbe is a BSL-3 microbe, right? It's not a BSL-3 microbe. It has been assigned a BSL-3 risk level, right? And it's sometimes a little bit arbitrary, sometimes not so much, right? But basically, these BSL or biosafety levels that we place on these microbes really indicate how dangerous it is, right? And there are 4 levels, going 1 from mildest or least risk to 4 greatest risk. And then each level of control is going to require certain amounts of PPE or personal protective equipment, a certain type of containment, and certain protocols that are going to be required to work with these things, right? So we'll start with BSL-1. Any BSL-1 organism is one that does not cause infection in humans, right? So for something like this, because it's not going to cause infection, we're talking about, minimal PPE. You can work on an open table if you want to. Any room in the world works as a BSL-1 lab. Sinks are nice because we should be washing our hands, right? But to be honest, not super necessary, right? And these are the type of things that you'd be comfortable with your little children playing with, right? Leaves. I know we don't think of plants as being like this thing that requires biosafety levels, right? But, it is a biological tissue, right? But plants, it's pretty safe, right? So these are generally assigned BSL-1. Even some microbes, which, are pretty much super low risk, can be considered BSL-1 microbe. BSL-2 organisms would be those that provide moderate risk to worker and community. And what that really means is that the worker can get sick, maybe someone outside can get sick, but it really only happens if you try to get sick. So it's often improper technique, improper protection, carelessness, recklessness. Those are the types of things that are going to happen. So these things are not going to jump at you. They're not going to float in the air for you to breathe. It's really lack of care and attention that's going to cause problems with these type of organisms, right? So as such, we have to build on what we just did, right? So you're going to need proper PPE this time, right? So wearing of gloves is very important. Maybe goggles if there's splash hazards. The rooms, BSL-2 labs have restricted access. You can't just have people come in and out, right? It's really irritating. We have BSL-2 labs here at Leeward, by the way. So it's really irritating when, the dean comes in and says, hey, I just want to check out your lab and see what's going on. I want to see a lab class in action. It's like, well, you're really not supposed to be walking around in here like this unless you yourself have proper PPE, right? Because people that just walk through, they can pick up some of these microbes and then they can get themselves sick or spread it outside, right? But again, it to be honest, it kind of takes effort. There has to be a certain level of biohazard control, so all the microbes that are growing, we don't just throw them in the garbage, right? At some point, they have to be destroyed or sterilized, and we'll talk about what that means, prior to disposal, right? And then if you can, you should have some sort of biosafety cabinet. I'll show you guys what a biosafety cabinet looks like and how they function in the next slide. Right, so there's these various levels that then make sure or ensures that all the organisms that you work with, all the microbes that you work with, stay in the lab. Right now, these are the types of microbes that if you were to carry a culture and you slip and fall and you drop your culture everywhere, not a big deal. Again, it doesn't go anywhere. A little bit of bleach, wipe it up. To be honest, the biggest risk is probably the glass shards that are created when you break a flask. But other than that, it's in general not so bad, right? BSL-3s, on the other hand, are microbes that can cause pretty serious, often lethal infection by inhalation, right? So these are the types of things that float around, right? So here, all of a sudden now, we need to make sure that the level of control is much more secure, right? So once again, we're going to talk about you got to have PPE. You got to have restricted access, right? In fact, it's often double doors to get in there, right? So it's like one door to get in where you scrub up and put your stuff on, and then door into the lab. Of course, there's going to be strict biohazard control, right? There's going to be not just, protocols before you start, protocols when you finish, but actually the organism that you use is also tightly regulated. So there has to be an inventory of the microbe that you use, whether it's a virus or a bacterium. It's carefully logged, right? So when you say, I'm going to use, let's go SARS-CoV-2, I'm going to work with that. He goes, well, okay, how much of it do you need? He goes, well, I'm going to take 3 microliters. So you actually, in your inventory, you have to indicate how much of that microbe that you took. And they're going to say, why do you need that much? Right? So, I think 1 microliter might be enough for it. You don't need 3 microliters, right? So all that stuff needs to be logged. You have to work in a biosafety cabinet. And then often you have to work with a respirator. Because again, these things can float around, right? There's going to be medical surveillance. So if you work in a TB lab, you're going to get regular TB tests to make sure that you're not sick, right? And then constantly, the airflow is also regulated. So even outside of these double doors, to get into the facility, it's what we call negative pressure. When that door opens, all the air gets sucked from the outside to the inside. Because in the event that there's some sort of outbreak, some sort of spill, which is going to be very difficult given that it's going to be, again, through double doors and whatnot, right? In the event some crazy thing happens and then the microbes may be able to escape the lab. Okay, they don't escape, but may be able to get outside the lab. The negative airflow makes it such that all the air goes in, so none of the microbes will float out, right? So everything stays contained, and then you do the whole disinfection control prior to anyone leaving. Okay? The the biggest risk microbes are BSL-4, right? And a BSL-4 are those that are easily transmitted, sort of, right? But really the key here is that they're lethal, right? So we have microbes that are extremely lethal and then often no treatments, right? So, maybe the most famous BSL-4 microbe would be Ebola because of the fact that it's, it's extremely lethal. There's no treatment. So if you get Ebola, you're kind of in trouble, right? You might as well say goodbye to your family because your chance of survival, very minimal, right? The BSL-4 labs are extremely tough to work in. Right, and it's super cool. These are the kinds of things that we see in movies, right? Look at this lady in her big bodysuit or her hazmat suit, right? But it takes like an hour to get in, right? Because you have to shower. So once again, double containment doors, right? And so you don't shower outside of the lab. You got to go in, shower through one of the containments. Then you got to put on one of these full body suits. And even though the suit is a pain to get into, you actually have to spend some time making sure that there are no leaks. So you put air into the gloves. Are there any leaks there? Air into the suit. You're constantly checking, right? Because a tiny little crack there, all of a sudden there goes in your microbe, and then now this person is breathing it, and then they're sick. The lab is self-contained, right? So it's containment within containment, oftentimes within containment. Very strict decontamination protocols. So the suit needs to be scrubbed. Your working area needs to be scrubbed prior to you leaving, taking off the suit, and then you got to shower, right? So it's really hard to get in and out, right? It's not something that it's, again, it looks super cool. It's not super fun. And the work needs to be done in this thing called a biosafety cabinet, right? So this has filtered air, right? So the air gets blown in from the outside, gets filtered, so then the air coming in is clean. And then right over here are exhaust vents and in the back as well. So then the air actually doesn't come out this way. It gets sucked down into there and then filtered prior to it leaving, right? So they're making sure that whatever you're working with, you get clean air in, clean air out, everything gets stuck in the filters. So then, of course, as the biosafety level increases, it's going to be more and more difficult. And as government regulated all these things, you actually have to approve for your BSL level, right? So, there's going to be less and less and less microbes labs, facilities for each BSL level. And sometimes it's really difficult to study microbes and pathogens because of the risk assigned to them, right? So, everyone has a BSL-1 lab. You are right now sitting in a potential BSL-1 lab. BSL-2s require a little bit more care, right? But for the most part, it doesn't require that much extra attention, right? So, every facility will have a BSL-2 space. You can create one in a high school. You can create one anywhere you want. This BSL-2 space, provided you take care of certain protocols and way of working. BSL-3, a little bit more of an issue. Again, a lot of it will be through cataloging. A lot of it will be through proper control, right? It does require certain more facilities. So there are hundreds of BSL-3 labs in the US, right? But only hundreds, right? So they're only, not 100, only hundreds. And then BSL-4 labs are so severe, and these are like multi-million-dollar facilities, right? $50-60 million to build one of these things, right? There's just a small handful, right? The CDC has a big facility. There's a big one in Montana, Galveston, Texas. So I think I'm gonna make this number up. There's like 5 or 6 operational BSL-4s in the country. And then I think there's talk of more that are being built or in the plans of being built, right? But currently, I mean, if you want to study Ebola and you want to work with Ebola, it's very small places that can do it. Very few people who are actually skilled enough to work with it, right? So, and that's why Ebola studies tend to be very difficult. Same SARS-CoV-2 is a BSL-3, right? It's difficult to do things like vaccine studies and pathogenesis studies and treatment studies because it's really hard to get access to BSL-3 labs. There are only hundreds of them throughout the country, right? So many times what you can do is you can do things like pseudoviruses where, you know, on the outside it'll look like SARS-CoV-2. On the inside, it's something that's safe, right? So then that will be then assigned a BSL-2, and you can do like these sort of studies. Okay. So we're now going to go through a series of definitions. Many of these things will sound very similar to each other. And oftentimes we'll use these terms interchangeably, right? But there are some very significant differences when referring to things, these type of terms. So we'll start off with sterilization. And what sterilization is, it's the complete removal or killing of, and the key word here is all microbes. All microbes will be completely either removed or killed. Right? This includes things like endospores, which are extremely tough to kill or destroy, as we have learned, and then viruses. Sidebar here. Viruses, remember, not living. Right? So we actually don't kill viruses because you can't kill something that is not alive. Right? So viruses are inactivated. Typically, this is done using high heat, pressure, or filtration, right? So filtration in this case wouldn't be killing of the microbe, it would be removal of the microbe. Most common is high heat, right? And then in this case, the sterilant would be whatever it is that we use to achieve sterilization, right? So then I'm going to rephrase my last sentence to say that the most common sterilant is high heat because we know how heat works. We know we can get it super duper hot, right? And we can get it hot enough to ensure that we kill even the most heat resistant of all hyperthermophiles. We know we can get there, right? Chemicals sometimes are used as sterilants, right? But chemicals are not preferred because the nature of how a chemical works makes it such that it's really hard to ensure that we're getting sterility, where it's really hard to know because microbes are all slightly different. As we learn, one of the major factors is this difference in microbes, right? And there's so many differences in microbes, it's hard to say with certainty that we're going to sterilize. Commercial sterilization is sort of like sterilizing. The idea is that we need to make sure everything is destroyed, right? But really, what we're talking about here are endospores. Now, oftentimes, commercial sterilization will end up sterilizing food, but it's definitely not guaranteed, right? So the purpose here for things like endospores, because especially in food, the most common food issues are things like botulism, right? And botulism is formed by an endospore forming bacterium. So it's always that endospore that causes the problem. And the purpose here then is to make sure that we retain food quality, right? We want to make sure that, of course, we want to get rid of the endospores, but we don't want to sterilize it such that we just completely destroy the food, right? And if you don't believe me, take like your canned soup or take some milk and you boil that thing for 20 minutes under high pressure, right? It's not going to taste as good. You're going to lose some of the proteins that give you the really nice mouthfeel to things, right? So you want to, get rid of the endospores, but still retain food quality. Now, maybe the question that we could ask ourselves then is, well, endospores are super tough. If things destroy endospores, shouldn't they destroy all microbes? Well, technically, yeah, right? But also endospores are going to be at a much, much lower level than microbes would be, right? So, there's this whole probability factor that goes into things. We expect there to be a certain amount of endospores. And then when we heat it for this amount of time, we're going to get rid of those endospores, right? But, bacteria may be present in greater amounts. So maybe we're not going to get rid of all the microbes, right? So it's this balance, this whole probability thing, right? So again, sterilization, complete removal of all microbes. Commercial sterilization is controlled at just all endospores, okay? So then lending ourselves to this, we have a certain way of working with things, right? So now we're talking about sterilization, right? So if we're going to work hard to sterilize things, destroy all microbes, we need to make sure that we use techniques that prevent the introduction of more microbes, right? And so this is what we call aseptic technique. Sepsis is this general term that refers to contamination. And in a clinical setting, sepsis refers to the presence of microbes in a patient, generally in areas where it's not normally found. And so when you get microbes growing in things like the blood where they shouldn't be, it leads to this whole systemic inflammation as the body's trying really hard to fight off this microbe that typically goes everywhere. And so that leads at the minimum high fever, but when it gets real bad, it leads to shock and death, right? So sepsis is kind of a bad thing, obviously, right? So when you're working with in the clinical environment or in the laboratory, we want to use aseptic technique. This whole idea that we want to make sure that we don't introduce microbes into these areas, into this sterile field, this area that's designated free of microbes, right? And this goes back to our friends, Lister and Semmelweis, who realized that it was the tools that we were using. It was the doctors who are working on patients that are ultimately going to lead to sepsis, right? And so it wasn't the patient, it wasn't this random thing that caused this patient to get sepsis. It was a doctor, right? So, aseptic technique then is very conscious of the fact, where can microbes be? And then how do I prevent this patient from getting the microbes from the environment, right? Because we, of course, do no harm. We want to make sure that we are curing and healing, not making things worse. Sometimes sterilization is not practical. It's really difficult to do. Sometimes it's not even really necessary, right? Because of the microbes present, there are only some that are really going to cause us some problems, right? So that's where disinfection comes in. Disinfection is using a disinfectant on a surface or fomite with this idea that we want to reduce quote unquote most microbes. And typically here, when we're talking about most microbes, we're talking about things like pathogens, right? So we have identified the problematic microbes, whether they are pathogens, whether they are just things that might spoil things, right? We know that these guys are going to be around, right? So what can we do to ensure that they're not going to be present, right? Now, we can't guarantee sterility, right? So disinfection is not sterilization, because some microbes may be around. We may still have endospores. Maybe some viruses are present, but we don't care about these guys. Maybe all we care about the viruses and not so much the bacteria, right? So we can't guarantee sterility, right? It may achieve sterility, but it's not about things being sterile. It's just getting rid of the microbes that really cause us problems, right? Antisepsis is similar to disinfection. It's the same as disinfecting, except it's all about treatment on living skin or tissue, right? So it's all about living tissues, right? So you can actually have a disinfectant. So something like alcohol, for example, is a disinfectant that you can use on a table surface, right? You can also use alcohol on your skin as you're cleaning things, right? Treating wounds. In that case, it would be an antiseptic. Certain things are good disinfectants, but bad antiseptics because they're very harsh, right? So bleach, for example, we often use bleach as a disinfectant when you're wiping on tables, right? You don't use bleach on your wounds because of the fact that it's very irritating on the skin, right? And then vice versa. Sometimes we use certain things that we use on the skin. Maybe they're not harsh enough for the table or the environment. Maybe they don't work as well on these surfaces, right? So, level of control is the same. Most microbes, particularly things like pathogens, right? But where you use it will determine the type of control that it is, whether it's disinfection or antisepsis. Degerming is often related to that. And sometimes degerming feels like you're using an antiseptic, but it's not actually the same thing, right? So in degerming, what you're doing is it's a physical removal of microbes, right? So all you're doing is you're removing microbes. You're not necessarily killing them. Maybe you can, but you're not really, right? So when you get your flu shot, you get the little injection, right? And they take the little alcohol swab and wipe that area. Purpose there isn't to use the alcohol as an antiseptic. Purpose there is just to push the microbes out of the way, right? Same with hand washing. When you wash your hands, right? Hand washing or soaps, they don't kill the microbes, right? So the whole antibacterial soap, useless, because soaps don't kill the microbes, right? All they do is they surround the dirt, they surround the microbes, and then when you rinse your hands, all the microbes wash away, right? This physical removal. And then finally, sanitization is removal of microbes to this kind of general public health levels, right? And this is kind of dependent on the public health department, whatever they feel is appropriate. And typically, these are the types of things that are shared, right? So most common, we're talking sanitization in restaurant, right? If so your dishware, utensils, those things are shared. Typically, you need to do something to remove the microbes to quote unquote public health levels, which then minimizes the transmission of disease, right? It's almost always done through high heat washing, right? So using soaps, hot water to wash. So it's this combination of degerming and disinfecting, right? Degerming and then kind of disinfecting, right, with the soaps removing, but it's mostly going to be, with the heat, right? But generally, we're not going to use chemical disinfectants on this kind of stuff. So you're not going to spray your plates with bleach and Lysol and then consider that a removal, right? Because we need to make sure that there's no disinfectant remaining. This is a chart from your book that kind of summarizes the levels of control, right? It's a nice little summary of what we just talked about, right? Along with some common applications and where we see these things, right? So it's a good way. And just most important here, make sure that you are understanding the differences between the words, right? They sound similar, right? But the definitions are very different. Finally, when we're talking, I guess not finally. So continuing on with microbial control, when we talk about levels of control, the types of control, we have cidal treatments and static treatments. In cidal treatments, we're talking about control that's aimed at killing microbes. In static treatments, we have control that's aimed at stopping growth, right? And sometimes we don't care. Either will work. Sometimes we need to have one over another. And sometimes, we need to have one over the other, right? So we can combine that then with what we're trying to control. So a bactericide, all right, bactericide that kills bacteria. A fungostat inhibits growth, right? And then, so again, sometimes why wouldn't we always want to kill things? Well, sometimes we want to preserve. Sometimes, the static control is less toxic, right? So many of these antibacterials or antimicrobials that are embedded in plastics, they tend to be of static control because, on a plastic surface in general, you're not going to have a lot of microbes, right? So just a few little microbes sitting around, that's more than sufficient. So that's totally okay, right? Whereas, if let's say you had a bactericide embedded, maybe over time that can start to build up some toxicity in the humans or the animals, right? Causing more long-term harm than is doing any good, right? And triclosan, or one of the antibacterials that were in antibacterial soap, is a good example of that. And we'll talk about that a little bit later. Oops, wrong way. And so when we measure microbial control, right, we can then see that just like growth, microbial death is constant, right? So microbes are going to be removed at a constant rate. So as such, we're going to plot this on a semi-log graph like we talked about. Super quick review. We have our linear scale time on the x-axis and then our logarithmic scale on our y-axis. And again, for those of you not super familiar or comfortable with exponents, right? Remember the 10 to the 0 means a 1 with 0 zeros. So basically 1. This here is a 1 with 1 0. So that's 10. A 1 with 2 zeros, that's 100, 1,000, 10,000, 100,000, etc., 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 right? And then this log graph really allows us to then, or the semi-log graph really allows us to see equal levels of decline, right? Because if we see here the decrease from 10 to the 10 to 10 to the 9, that's 10 billion to 1 billion, right? This decline is exactly the same as here, which goes from 10 to 1, right? So we can still see that from 10 to 1 as a decline. Same with 10 billion to 1 billion. Now you might think, why those numbers aren't even relevant to each other. 10 billion is like ridiculously large, and 10 is super duper small relative, right? But really, if you look at it from a percentage standpoint, this 10 to the 10 divided by 10 to the 9 is exactly the same as 10 to the 1 divided by 10 to the 0, 90% reduction. Okay, so what we're looking at it with, and often we'll say a 1 log difference, we're looking at a decrease of 90%, right? And so sometimes when we're talking about microbial control, we refer to something called a decimal reduction time, right? Just the time it takes to reduce a population by 1 order of magnitude, by 1 log, by 90%. Okay, so let's look at this graph then. Here we have 10 to the 6, that's at 20 minutes, 10 to the 5, at 25 minutes, right? So for 1 log difference, or 1 order of magnitude, or 90%, it takes 5 minutes. And then we'll know, now that we know that, if I say, well, how long does it take to go from 1,000 to 100? Well, here's 1,000, there's 100, 5 minutes, right? So this is very controlled. So then if I say, okay, well, we have this population, now we have 1 million, and I need to make sure it gets down to 1,000, because that'll be sufficient for me, how long it takes to get there. And because it's going to be constant, we say, hey, we lose 90% every 5 minutes. That means we can go 1, 2, 3. Okay, so that's 15 minutes it takes to get from 1 million to 1,000. All right, so what then makes a certain control effective, right? Well, there are many factors that go into it, right? So some of the major things that you always have to consider when dealing with any type of microbial control are these various factors, right? Time, the longer the better, right? The more exposure there's going to be to your chemical or your sterilant or whatever treatment that you have, the better, right? So we say heat is important. Heat is an excellent sterilant, right? But time is an important factor of that. So you can't just take some boiling water, some steam, and just like flash it over something and say, well, look at that, I just sterilized the surface because steam is hot and it's going to kill everything. Well, like a 1 second exposure isn't enough, right? You actually need a certain length of time. Everyone can dunk their finger into boiling water for a second, right? Don't do that, please. But everyone can just really quickly in and out, right? And then you'll be fine, right? No damage, right? Now you keep that in there for 15 minutes. Well, guess what? Damage, right? So time is super important. Microbes themselves are going to be resistant to certain types of treatments and much more prone to certain types of treatments, right? So envelope viruses, for example, their level of susceptibility is going to be way different from non-envelope viruses because of the nature of the structure of the virus, right? Same with microbes, gram-positive bacteria, gram-negative bacteria. They're all going to be affected differently. The concentration of the sterilant is important. So if you have a certain chemical, higher concentration, the better, typically, right? But there's always going to be this balance, and we'll talk about that a little bit later. And then, of course, the environment, right? So if something is extremely wet, maybe that might inhibit some of the sterilants. Definitely when you're talking about chemicals, so stuff like organics, vomit, and blood, they often interfere with chemicals working. So if you have to clean up vomit, you can't just disinfect it. You actually have to clean up the vomit first and then disinfect after, because the vomit and the stuff that's in there will inhibit the disinfectant from working properly. Okay, we're about half hour in. Now's about a good time to take a break if you want to, right? Grab something to eat, just stretch your legs a little bit. If you want to keep going, you can keep going. If you want to take a pause, right? I'm going to keep going. We are going to talk about 2 general methods of microbial control: physical methods, chemical methods. And of course, each of these are going to have different methods of application, different advantages, and then different factors to think about. And then the most common physical method is heat. Right? It kills by, as we learned, it denatures proteins. Once proteins are denatured, cannot metabolize, right? If you and it destroys membranes, right? Because membranes are very sensitive to these types of things, right? So we all know, make things hot, things will die, right? We can then define a microbial culture or a population based on two types of measurements: thermal death points and thermal death times, right? So a thermal death point implies that we're going to keep a same length of time and increase temperature, right? And a thermal death point then would be the lowest temperature at which all microbes are killed, right? In this case, at which all microbes are sterilized, right? It doesn't have to be 10. We can do it at 5 minutes. We can do it at 15 minutes, right? But whatever the case may be, thermal death point then would be just the lowest temperature it takes to kill, and which then implies the higher the temperature, the thermal death point, the more difficult this population will be to kill. And it can be because the microbe may be more heat resistant. It may form endospores. Maybe there's just more of them. Many bacteria are going to require more time or a higher temperature to sterilize. Thermal death time is related to that. That's the length of time needed to kill all microbes at a certain temperature. Right? So we have now a constant temperature, and then we're going to keep the microbes in there for a certain time. Right? And once again, certain microbes are going to have different thermal death times based upon the number of microbes present, right? Based on the characteristics. So in the lab, it's often visualized as something like this, right? So these things are growth, right? So that's how we visualize growth. We have two different bacteria, Bacillus subtilis on this side, E. coli on this side. I believe the temperature here was 65 Celsius. And then the cultures were incubated for 0 minutes, basically not at all. 1, 3, and 5, 0, 1, 3, and 5, right? And we can see already differences in our thermal death times, right? So what's the thermal death time of our E. coli culture? If you said 3 minutes, that's correct. Not 1 minute, right? So it's not the last time we see it, it's the first time we see it gone, right? Whereas our Bacillus subtilis culture, our thermal death time, right, 5 minutes. So in this case, we would say that Bacillus subtilis has a longer thermal death time than E. coli, which means that it's more difficult to kill. Now, why would that be? What is it about Bacillus subtilis that makes it more difficult to kill, right? Well, in this case, it's because Bacillus subtilis is an endospore former. So overall, it's going to be more heat resistant than E. coli. Good? All right. So let's talk about types of heat. Because, yeah, it turns out there are different ways that we can apply heat, right? Maybe the easiest is something called dry heat, right? We just pretty much burn it, stick it in the fire. So in this case here, we have a loop. This inoculating loop is used to transfer bacteria from one thing to another thing, right? So by sticking in the fire, getting this extreme orange glow, we destroy the microbes in there, right? We can also stick things in a burner or an oven. We can stick things in an oven, right? A dry oven. Typically, sterilizing time would be 170 Celsius for 2 hours, right? In that case, everything will be hot, and then the microbes will be completely killed. We would say we've sterilized whatever we stuck in the oven. Moist heat sterilization is the application of heat and steam, and it's much more effective than dry heat because the heat has this effect of kind of bunching up or bundling the proteins, which then makes the heat so much more effective. And typically, so when we talk about equivalent treatments, using moist heat, the equivalent treatment will be 121 Celsius for 15 minutes. So we say this would sterilize. Whereas for dry heat, it's 170 Celsius for 2 hours. Right? Both of these achieve sterility, but look how much easier it is using moist heat than dry heat. Right? Because of the fact that, again, the addition of the steam really causes it to be much more effective. There are other types of heat treatments. So pasteurization, named after our good buddy Louis Pasteur, one of our founding fathers of microbiology, is a limited heat treatment to reduce spoilage organisms, right? And again, the story was that, he recognized that microbes cause disease, and microbes cause disease. Oh, sorry, the other way around. So he recognized quickly that food spoilage was due to organisms, right? Some sort of microbe within the food, right? And then he learned over time through trial and error that he can destroy or at least increase the shelf life of the food, right, without destroying the quality of the food. And of course, the food that he worked at was wine, right? Because he's French. Wine is so important to French culture, right? Kids need to drink their wine. So It's all about making sure that you can get the wine to these great distances because they need to stay a little bit longer, right? However, without destroying the flavor of the wine. Back in the day, it was 63 Celsius, so not boiling, right? Just warm for 30 minutes. And that was sufficient to make sure that you were retaining the quality. Modern pasteurization is extremely much more effective, right? So HTST is high temperature, short time. Typically, your milk is HTST pasteurized at 72 Celsius for 15 seconds, right? And then we bottle it and refrigerate it. Sometimes you'll see in the stores UHT, ultra high temperature, 138 Celsius for, I think it's like 1.7 seconds or 1.8 seconds, right? And then sealed in a container. And these things will last much longer. Sometimes you'll even see these things on the shelf, right? It's like, hey, here's milk on the shelf, not in the refrigerator, which is super weird, right? All right, that's high heat. High heat is cidal. It's going to kill, right? Low heat is static, right? So we can then refrigerate and we can freeze. The lowering of the heat will slow your metabolism. Ultimately, it helps preserve the samples, right? Whether it's the food that you want to eat for later, whether it's the microbes that you want to store for later, right? Now, freezing can kill some microbes, right? So some, especially eukaryotic microbes, they tend to be really sensitive to things like freezing and thawing, right? So sometimes a slow thaw helps preserve the samples a bit better. Right. And then typically if you're going to do long-term storage, we use super low temperatures, right? So typically your home freezer will be about minus 20 Celsius. Our ultra low freezers are going to be around minus 70, minus 80 Celsius. So super duper low, right? And then again, here the idea is that we'd like to preserve our samples, static control, as opposed to killing, right? Another physical treatment is pressure. It's not super duper common, but it can be used. So Pascalization is the processing of food using high pressures and things like pressure cookers, kind of like a pressure cooker, right? Again, the idea is that it helps extend shelf life, maintains food quality, right? It is a cidal control, but generally endospores can survive it. So this is not the type of thing you would use for stuff that you may suspect endospores to be in. And then we can even use this for treatment of humans. So in a hyperbaric chamber, what you do is you increase the oxygen and the atmospheric pressure. This extra shot of oxygen really helps, your body work a little bit more efficiently. It really puts extra stress on microbes. It's not something you can do for too long, right? Because again, microbes are biological, so anything that affects them is going to affect us, right? But it is something that can be used for limited times in clinical infections. Desiccation or the drying is a static control. And we talked about this, right? AW, do you guys remember what AW stands for? Water activity, good, right? And what is the water activity of, or AW of water? 1, right? Now, when we desiccate things, we reduce that, right? We want to make sure we get it as low as possible in order to prevent any water being around. And then this then would inhibit metabolism. Right, and we talked about this with respect to like bacteria, which need a, really they don't metabolize well, lower than 0.9, 0.8 AW, right? But then, what type of microbes can tolerate even lower than that? Fungi, right? So we talked about how fungi, they're actually capable of tolerating much lower water activities, right? So when you're making your own home jerky, make sure you gotta really dry it. Not dry enough, really dry it. Related to this is lyophilization. Lyophilization is freeze drying, and it causes much less damage, right? And then, which then actually means it can last for much longer, right? So, famously, the astronauts took freeze dried food up with them, right? So you have this food product, you freeze dry it, it lasts pretty much forever, right? Because you have this double effect. It really, it draws all the moisture out. If you've ever had freeze-dried food, it's like flaky, right? And then we can actually use lyophilization to preserve microbes as well, right? But it's used for many, many things, foods. When we transport things like medication, we can lyophilize it. This way we can keep it on the shelf. It doesn't have to be cold until you rehydrate it using water. Radiation is another very common method of control. Radiation are wavelengths of light smaller than visible light. All right, so it's used to kill, and it's a cidal treatment. It uses to kill two kinds of radiation, ionizing radiation and non-ionizing radiation. Ionizing radiation is characterized by its real power to penetrate things, right? So it's very intense. It passes through things, right? And it kills things by damaging cell components, right? So it passes through cells, really destroys their DNA and their membranes. Super short wavelength kind of stuff, right? So, we were familiar with them as, X-rays. What do X-rays do? Well, we use them to take pictures of inside of us, right? So again, deeply penetrating. Gamma rays typically is what used to sterilize things like, stuff that doesn't really autoclave very well, right? So things like plastics, which would melt, food, which would get destroyed, right? So actually it's commonly used in the food industry, more so in Europe than here, right? But definitely some of the things that do get processed through Europe, if you ever see a symbol like this, it means that it's been treated using gamma rays, right? And it's so easy. You just take the package, you put it through the big gamma ray machine, it just like zaps it, right? And then it comes out nice and sterile on the other end. Careful though, it's extremely sterilizing, right? I know in comic books you think, oh, I'm just gonna go walk through, get some gamma rays, become this incredible Hulk type of thing, right? And the reality is, no, it would just pretty much destroy you, right? Non-ionizing radiation, also cidal, right? Little bit less penetrating. So really how it works, non-ionizing radiation works by destroying DNA, and it does so by taking these thymines that are next to each other, what we call adjacent thymines, and it forms these thymine dimers, right? So it forms this covalent bond between these two thymines. Now we do have repair mechanisms to be able to fix this. So we have these enzymes that'll come, and then basically it removes this chunk of DNA that has these thymine dimers and replaces it with proper non-thymine dimered DNA, right? But you start to create thymine dimers everywhere, and at some point or some rate, they're not all going to be fixed. So then these non-ionizing radiation are going to, at best, kind of remove some small amounts of DNA. At worst, we're going to lose DNA all over the place, such that it's like so damaging that the cell just ends up dying, right? Examples of UV light, of non-ionizing radiation, examples of UV light. What I'm saying. The examples of non-ionizing radiation is, how come you're not working? There we go. UV light, right? So UV light is something that we use oftentimes in certain areas, right? So especially in lab bench and sometimes hospital rooms where we have this really flat setting, shine the UV light, destroys everything, right? But very important, it does not penetrate very well, right? So something like as simple as plastic, sunglasses, it doesn't penetrate. So UV light is unable to get through these types of things. So when you're sterilizing the surface, you see this area under the flask, not sterilized. This area under this tube rack, under this, not sterilized at all, right? So you always have to be aware of when you're using it. It's much easier to use. I mean, if you were to use gamma rays on this thing, it would sterilize the whole thing, right? But the level of containment for gamma rays is much more severe, right? When you think about, you go to the dentist's office and they say, okay, we're gonna go and take an x-ray. It's totally safe. You just sit here. I'm gonna leave the room and hide behind this wall, right? But don't worry, it's totally safe, right? Because of the fact that it's so penetrating, you got to be careful, right? And you got to wear that thick jacket. So again, each of these are going to have their pros and cons. Filtration is the last physical method we're going to talk about. And here it's really just removing microbes using filters, right? Typically air is filtered. We can filter liquids, but liquids not so much for a constant type of thing, right? Liquids we often filter and then have to change the filter, right? Whereas with air, we just like blow the air through. And then the most common application is using a HEPA filter, which stands for high efficiency particulate air filter. Typically the pore sizes are on the order of 0.1 micrometer, 0.2, sometimes 0.5 micrometer. Really small enough to trap microbes. Some viruses can get through. Depending on the pore size, right? And so the air coming out is, we say, nearly sterile. Really depends on the level of the filter. Definitely microbes won't be present. Mold, dust, all that kind of stuff won't be present. Maybe some virus will slip through. So again, in your book, there's a nice chart that summarizes what we just talked about. Um, Again, you don't have to memorize this, right? But it is a really nice overview of what we just talked about, and then some examples of how we use them. We didn't talk about all of them. So, example, sonication, we didn't talk about that. So, if we didn't talk about it, you don't have to know it. All right. So, when it comes to chemical control, things are a little bit different here now. Chemicals are so different. Microbes are so different. There are many things to consider when using chemicals. So, first and foremost, what type of microbe are you targeting? That, of course, includes, gram-positive, gram-negative bacteria, but also, the difference between bacteria versus a virus versus a protist versus an algae. What do you expect to be there? What's going to cause you the most problem? Cleanliness items. We've kind of talked about this already. Just with respect to how certain types of organics can interfere with how well chemicals work. How is the item going to be affected? So if you have, let's say, a very porous environment, maybe you're not going to want to use a liquid chemical. That's definitely going to affect a certain thing that you want to use, right? So, and then of course the safety of the chemical itself. If this is going to be something that humans are going to touch a lot, a lot of skin is going to come into contact with that. You definitely can't use something that's too irritating, right? But if it's a bench table, doesn't really matter. And then certain things that we don't consider too much are cost and the ease of use, right? If something is extremely effective but requires a lot of work to use, you need to put in a certain aerator, right? Or it's just really expensive. Sometimes, maybe we can use this other thing, which is just spray on and wipe. It may not be as effective, but it's super cheap and very easy to use as opposed to like the super heavy duty stuff. Okay, so we're gonna go through and just talk about some common chemicals, a little bit about how they work and maybe some of their applications, right? As we go through this thing. Primarily, I just want you guys to understand just a little bit about the class of these different types of chemicals. And we're gonna start off with phenolics. Phenolics were probably the very first major disinfectant / antiseptic used. Hey, you should know what the difference is, right? The disinfectant versus an antiseptic. And this goes back to Joseph Lister, who used carbolic acid as his way to antisepticize surgical wounds and all that kind of stuff, right? Phenolics are characterized by this ring right here. This benzene is what we call it. It's a hexagon with these double bonds here, and then this hydroxyl group sticking off that, right? And phenolics are nice because they're very stable, they're persistent, right? So they're very good mechanism of action, right? They work by disrupting membranes, denaturing proteins, and so commonly they're used as topical detergents in clinical settings. And triclosan, which is an antibacterial soap, is also very common, right? However, one of the advantages of phenolics, the fact that it's very stable and persistent, actually makes it a little bit challenging to use because of the fact it does persist, right? So, over time, it does cause some lingering issues, or can, right, if you don't clean it off after use. But more environmentally, triclosan, it just kind of persists in the environment for a long time. And, there are studies that have shown that bacteria can develop resistance to triclosan because of the fact that it just kind of sits around in very low concentrations as it gets flushed down the drain, right? So actually, because of that, it's now been banned for use in antibacterial soaps. So you really shouldn't see it anymore. And if you ever do see it still in antibacterial soaps, it's an old product that's kind of persisted. But the FDA have definitely said no longer appropriate for use. Do I want to say this? Yeah, why not, right? So Lister, I may have mentioned this already. Oh, shoot, I'm getting to that age where I feel like I'm repeating stories. But Joseph Lister, he kind of lent his name to Listerine, right? So the people that invented Listerine, even though there are no phenolics in it, right? They initially developed Listerine for a surface disinfectant and an antiseptic, right? And then so in homage to Joseph Lister, who kind of developed this whole idea of using antiseptics, they called it Listerine because of that. Heavy metals, not the music, right? But literally metals that are heavy on the periodic table. have been used for antimicrobial properties for centuries, right? So they kill by binding proteins, right? And it inhibits enzymatic activity. They just kind of get in the way of many of the cofactors that are present, which sort of look like some of these heavy metals. And then just stop them from working, right? And so we say the action of heavy metals are oligodynamic in that you don't really need too much. Small concentrations can have quite a significant effect, right? Because of the fact that we don't metabolize them. They just kind of stick around. So they kind of accumulate within cells, right? So over time, these things are going to cause some problems, right? Which then, of course, makes an issue. It's not selectively toxic, right? So humans, it'll accumulate in humans as well, right? So if you're going to go and let's say make some sort of treatment or therapeutic that has these heavy metals, right? Over time, it can accumulate in you and then definitely cause issues. This person here is suffering from silver poisoning, this weird bluing of the skin. And this bluing of the skin is just this accumulation of silver over time. Who knows? Maybe this person had like silver fillings or I don't know what. Drank it. Who knows, right? And actually, there are some treatments, colloidal silver, a little different. It's not quite the same, right? But you can drink that. Generally, I always say I would never drink it because it's, because of this whole not selectively toxic, but maybe the whole colloidal silver part is different. I don't know. They still use silver as in combination with antibiotics because it often helps make their efficiency, right? Sometimes they'll lace it in bandages, right? So it's still used occasionally, right? But again, it's not something you want to use for a long time. And then things like, copper, nickel, zinc, many of the old utensils and things like that were made from these heavy metals because of their antibacterial properties, right? So, they do have an effect. They're definitely useful for its application, right? But again, the big issue here, not selectively toxic, and then definitely can have an effect over long periods of time. Good. Another very common group of disinfectants or chemicals, are the halogens. And halogens are, if you look at the periodic table, it's the second last row on the right, right? And these are our gases: iodine, chlorine, fluorine. We know chlorine, right? It's in bleach, kind of. We're gonna talk about that a little bit, right? Iodine has been used traditionally a lot for antiseptics, right? Typically what we call a tincture, right? This alcohol, mixing it with alcohol helps make it more effective, right? It mobilizes more as it keeps it more soluble, or as an iodophore, which is iodine stuck to an organic molecule. And it looks like this. I don't know if you care. Fluorine, we know all about fluoride, right? Fluorines have antimicrobial properties because it interferes with their metabolism, right? Helps some of their synthesis of things, right? So we use that for preventing cavities, and generally these things, in the right doses are not really an issue for us, right? We see halogens more in the form of chlorine, which is a strong oxidant. Typically we're going to see it as this hypochlorite, right? And so that's the active compound in bleach, right? So when we say things are chlorine, it's actually the chlorite ion. And then now more for a lot of cleaners, chloramines are what's used, right? So this is ammonia, and then you attach a chlorine item over there. That's a chloramine, and they're very common for cleaners and disinfectants, right? Now, as we know, with the example of bleach, some chlorinated compounds can irritate the skin and the tissues, right? So this is not something that you're gonna apply on the skin for long periods of time or too often, right? Maybe some short immediate use, right? Generally, you're gonna apply this on some sort of surface, right? Some sort of table or floor. And then, you do have to clean it off, right? And then there are some microbes that are able to resist chlorinated compounds. Alcohols, we're all familiar with that, right? Alcohols work by denaturing proteins, disrupting membranes. The interesting thing about alcohol, typically we say more is better, right? So, we want a certain concentration. We actually talked about how the concentration of your chemical is important, right? However, it actually requires a little bit of water to be effective, right? So there's this range of alcohol that's useful. So somewhere around 50 to 60%, it starts to become useful. And then right up until about 80 to 90%, right? 100% alcohol is actually not very effective at all, right? So that's why when you see hand sanitizers or alcohol from the store, right? They're typically at 70%, 60% alcohol, 100% useless, right? So if you ever do get your hands on 100% alcohol and you want to use it as an antiseptic, right, or a disinfectant, make sure you dilute it some, right? Mostly alcohol, some water. Useful because they're good bactericidals, they're fungicidal, but only for virucidal effects, only for envelope viruses, right? Because the envelope is important for the survival of the virus. Did I say survival? Remember, you can't kill viruses. So it's important for the virus to remain active, right? Or infectious. If you can strip that membrane away or the envelope, the virus now becomes inactivated, right? So it's very useful against multiple types of microbes, right? It does not, however, eliminate endospores. Now, good thing about alcohols, they're cheap, right? However, because they evaporate so fast, it doesn't last very long. So it's one of these things where you spray it down, you wipe, right? Let it do its course of action, right? And if it's something that's particularly requires a lot of disinfection, you need to spray more or multiple times. All right, surfactants, right? Surfactants are comes from the term surface active agents, and they're the soapy things in soap, right? They're amphipathic molecules, this idea of hydrophobic tails, hydrophilic heads, right? And what they do is they form these micelles, these emulsions with dirt, surround them, right? Remember those micelles, right? Where we have our circles of lipids that surround things, and then the water will just simply wash that away as you rinse. Now, for surfactants to work, it does require sudsing, right? So if you ever put on soap and then it doesn't sud and you rinse it before before it starts to sud, it's not working, right? So in many ways, the best way to think about it, it's the bubbles that work, right? It's the bubbles of the soap that surround the materials, like the bacteria, like the dirt. And then you got to wash the bubbles away. And as you're washing the bubbles away, it'll wash away with it the things that it's surrounded, right? So in areas where there's hard water, and we have a little bit of hard water in Hawaii, but not too much, right? But in areas where there is hard water, and it doesn't sud very well, surfactants don't work. So typically they'll need to use different soap formulations in order for soap to do what it's supposed to do, right? So, but always remember, it's the bubbles that do the work. When you use soap, make sure you get yourself a really nice lather and lots of bubbles. I don't think this is finally. No, we still got a few more, right? So quats or quaternary ammonium salts they look kind of like phospholipids, right? Which then is really their method of action. Pyridinium, very common in mouthwashes. So if you ever look at the active ingredients, you'll see, look at this thing. It looks kind of like a phospholipid, right? A little bit different, a single tail. So what they do is they insert themselves into plasma membranes and then they start to disrupt them and then causes them to fall apart, right? Which means then it's nice and active against bacteria, against fungi, protozoans, envelope viruses, but not non-envelope viruses. And the nice thing is it's stable, it's cheap, generally non-toxic, right? So, at the levels that they appear in our mouthwashes, for example, generally it doesn't cause us too many problems, right? So it's a really nice chemical to use. Bisbiguanides, these are positively charged antiseptics, which then means it attacks the negatively charged membranes, disrupting their cell membranes. Once again, anything with the membrane then can be affected, like bacteria, like envelope viruses. The nice thing about this is that you can use these in different concentrations, and at lower concentrations, they tend to be static. At higher concentrations, they're cidal, right? So you can kind of adjust according to your needs. Typically used in surgical scrubs, oral rinses, right? Often in combinations with quats and various other things. Alkylating agents are disinfecting chemicals that work by adding alkyl groups to molecules. And alkyl groups are just these carbon things here. So this CH3, for example. They often work by attacking DNA, right? So here we have guanine. And then so the guanine alkylating agent will add an alkyl group. Oh, sorry, it's right over here. So here's guanine, and then here this is ethyl guanine because it's got this. These two carbons are ethane group, right? So it's been alkylated, and then that then starts to affect, of course, DNA structure, which then starts to affect, which then will eventually lead to cell death, right? Because of the fact that endospores also have DNA, alkylating agents work really well against endospores. However, because of this kind of broad nature of things, we often use them as sterilants and then typically to preserve specimen, right? Because, we're not going to take these things internally. This will be an external use only. And then finally, I think this is the last group of chemicals. We'll talk about the peroxygens, aka the peroxides, right? So, and we often see this in hydrogen peroxide. And we talked about how the peroxide anion is a reactive oxygen species, right? So it's going to go and do its thing by just really attacking cells, attacking the DNA, cell membranes, proteins, and all that kind of stuff. Super cheap. It's safe for the environment. It'll naturally break down into water and oxygen, right? So if you just leave hydrogen peroxide out, eventually, if you ever open an old bottle and you hear a right, that's the oxygen that's built up and it starts to leak out, right? And if that's a really big, you pretty much don't really have hydrogen peroxide left, right? So in that case, dump it, time to get yourself something new. And generally broad spectrum. It does work on endospores, but it takes a little bit longer for endospores. All right, but it's a very nice alternative or a very nice option for, if you need something that's relatively cheap and safe. It's not very harmful to us as well on the outside, right? So that's why it works excellent as an antiseptic, because it doesn't irritate the skin on most people. All right, so let's talk a little bit about food preservatives, right? Chemicals, of course, are used to prevent spoilage. I don't want to talk too much about food preservatives. There's so many. The food industry is obviously a huge industry, right? And it's all about food safety, right? So I don't want to talk too much about it, right? But just if you look at some of these things, sorbic, benzoic acid, propionic acid, right? All of these things tend to inhibit enzymes by generally lowering the pH of the environment that the food is in. The nice thing about propionic acid is that it actually works very well at higher pHs as well, right? So you can use this in different types of foods that are not necessarily acidic. Sulfur dioxides, or sulfites, as most people see them as, are used in dried fruits, right? They help with this oxidizing effect, right? So the microbes are inhibited, right? Problem with sulfites is that some people have allergies to sulfites, right? So for them, doesn't work out too well. And same with and often you'll see nitrites, a little different from nitrates. The main purpose of nitrites is to stop the germination of endospores. Common in beef or in meats, packaged meats, so you'll often see that in the form of sodium nitrite. One issue with nitrites is that when you cook it, sometimes you get these nitrosamines that are produced, which have been shown to be carcinogenic. Again, small amounts is not a big deal, right? So any of these things that you take, they're tested, they're called GRAS, generally regarded as safe, right? But the idea is that over time, if you eat a lot of these things, right, it can accumulate and over time cause some problems, right? Which is why I always say, you should enjoy what you enjoy, but never try to overdo anything, right? So even like bread, which we love, or, fruits, yeah, it's great. But if you're going to eat these things all the time, well, that's not good for you, right? Vitamin C, don't eat too much of that stuff. Not good for you, right? Everything in moderation. And then finally, with respect to food preservatives, there are natural food preservatives. So many microbes, actually the word antibiotic is a term that refers to an antimicrobial that's produced by a microbe, right? So there are actually many antimicrobials that are produced by microbes that produce our food, right? So Lactococcus lactis, for example, produces this thing called nisin. And so many of the cheeses and things like that have nisin in it that are produced by Lactococcus lactis, or sometimes they will extract the nisin and then use that for various other products like meats, right? So nisin is simply a natural antimicrobial. Same with natamycin. That's in many food products, especially dairy products, and they're used for antifungal properties, right? So once again, this is something produced by a bacterium, Streptomyces natalensis. All right, almost done. We're going to talk a little bit about how do we determine and measure the effectiveness of an antimicrobial. The very early measures of how good a chemical was called the phenol coefficient. And phenol kind of became the standard for comparison because it essentially was the very first, most well-used antimicrobial. Basically, you take two bacteria or whatever test bacteria that you want. Typically, it was against Staph aureus and Salmonella typhimurium, right? But any bacterium that you want, and then you expose it to a chemical for 7.5 minutes, and then you just compare that to phenol, right? Whereas phenol is 1, right? So if it's the same as phenol, then you would say that's 1. If it's lower than 1, it's not as good as phenol. If it's higher than 1, it's better than phenol, right? So for example, against Staph aureus, formalin has a phenol coefficient of 0.3, which means it's probably not something you want to use. The Staph aureus is the microbe of interest, whereas chloramine has a phenol coefficient of 133. So if you're going to use, or Staph aureus is the microbe that you're concerned about, chloramine would be the better choice. And warning, here comes some more math. All right, so to calculate a phenol coefficient, it's pretty straightforward. All you would do is take, after you dip your test bacteria and then you grow it onto some agar plate, basically you would just count the number of bacteria, right? So you'd have a plate like this. How come you're not working? You have a plate like this. Bang, 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 right? And you count, and then you'd say phenol. After treatment of phenol, you'd have, let's call it, 10 colonies, right, or CFUs. And then we have our test X. We would have, let's go, what's going on here? We'd have 30 CFUs, right? And so, just even thinking about this, you got to think to yourself, okay, after I treat it with phenol, I'm left with 10 CFUs, right, or quote unquote 10 cells. And then after treating with this treatment X, we have 30 CFU. So you should be able to see, hopefully, that X is worse than phenol because we still have more bacteria left, right? But then we can turn this numerically and simply just 10 divided by 30, and that'll equal to 0.33, whatever you want, 33 phenol coefficient, right? So it's relatively straightforward, and then so it's always compared to phenol. Another very useful test is called the disk diffusion method. It's at its simple. The principle here is that you take your antimicrobial, you lace it into a disc, right? So sometimes you buy these things from the manufacturers with various antibiotics. Sometimes you can just take a cotton or filter paper disc, you soak the chemical in it, and then you place it on this plate that has this agar plate that has bacteria on it, and then as the chemical diffuses away, the growth of the bacteria here will be inhibited, right? So if the chemical is especially effective, right, the zone here that we see, or the zone of inhibition, will be larger. So a larger zone means that the chemical is more effective, whereas if it's less effective, you're going to see a smaller zone of inhibition, right? So we can then use that to test the effectiveness of a chemical against a specific bacterium. We can also see the effectiveness of a chemical generally across the board, right? So here we have two bacteria, Pseudomonas aeruginosa, Staphylococcus aureus, and then what's going on here? Okay, there we go. And then Staphylococcus aureus, right? There we go. Pseudomonas aeruginosa, Staphylococcus aureus, and then in general, 1, 2, 3, 4, 1, 2, 3, 4, right? In general, you can see here that Pseudomonas aeruginosa is actually very resistant to whatever these test chemicals are, right? So this one here has a zone of inhibition, a zone where there's no growth, right? These three here had zero effect, right? Whereas for Staph aureus, you can see that these zones, all of these chemicals were effective against Staph aureus, right? And then the zones in general, if you're looking at comparable, notice how much larger this zone is compared to this zone, right? So in general here, it looks like that Staph aureus is much more susceptible to chemical attack than Pseudomonas aeruginosa is, right? And the reason for that, in part, is because Staph aureus is gram-positive. In general, gram-positive tend to be more affected by chemicals because the gram-negative bacteria are able to resist the chemicals because of their membranes. Their outer membranes are able to kind of bounce off some of those chemicals much more effectively than a gram-positive bacterium, which has the peptidoglycan right on the outside. This is a very nice test. Sometimes, by the way, it's called a Kirby-Bauer test, right? This disk diffusion or Kirby-Bauer test is easy to perform, right? So it's a very quick way to determine whether a certain chemical or how effective a chemical is against multiple types of bacteria, right? But sometimes it has its limitations, right? So for example, if you want to see how effective a chemical is on a surface, you would use something called a use dilution test. So in a use dilution test, you take a steel cylinder, you place it in a culture, right? Basically, you're now lacing this steel cylinder, this thing right over here, with bacteria, and then you let it dry on there. Once the bacteria have dried on these steel cylinders, you then place it into the chemical, whatever it is that you choose, right? For a certain amount of time. Then you take it, place it into fresh growth media, and then if you see growth, that means the bacteria on the cylinder have survived, right? Which then means that the chemical was not effective, right? If the chemical was effective, you will see no growth, right? There's a very strict protocol for doing this, right? It's typically used for things like clinical chemicals, which are used a lot on certain type of surfaces, right? And then from that, you will then determine how effective it is against certain type of bacteria, and of course, the length of time that you have to use the chemical for, right? Very common type of thing, oversimplified here, right? But it's definitely used a lot for chemicals used, many disinfectants. And kind of related to this is an in-use test, right? So a use dilution test tests for the effectiveness of a chemical on a surface. The in-use test determines if your chemical or your solution is contaminated, right? So, and this is something that has to be done in a properly managed clinical setting, right? So you have this chemical. Remember, you're going to have your various chemical. You're going to spray it. You're going to do stuff with it, use it, right? Every now and then, especially if you use a rag, sometimes the stuff that's on your rag gets back into the chemical, and you want to make sure that your chemical itself doesn't become contaminated, right? So what you do then is you take your disinfectant that you've been using, you take a little bit of that, mix that with a sterile broth that allows for bacteria to grow. Then you take some of that, place that on an agar plate, let it grow for a certain extent of time, and then you start counting the colonies, right? And if you have a significant amount of growth, in this case it's 5 or more colonies, then you would say that the chemical is contaminated, and then would have to be disposed, right? Whereas if you see no growth, you say, okay, well, the chemical is still okay, you can continue to use it. All right, that's module 7 for us, right? We learned probably a lot, hopefully, about how we can control growth, and how do we destroy bacteria. We learned a lot about the vocabulary for microbial control, subtle differences between sterilizing, disinfection, antisepsis, et cetera, right? And then probably more than you ever wanted to know about chemicals, right? But if you ever find yourself in a clinical setting, these things are going to be important, and you're going to definitely hear many of these types of chemicals coming up. And hopefully one day someone can say, hey, can I use these quats on in order to prevent SARS-CoV-2 from being here, or inactivate SARS-CoV-2? You would say, yeah, because CoV-2 is a enveloped virus, and quats work on enveloped viruses, right? Okay, that's the end of module 6 for us. We will see you in module 7, in which we will talk all about the DNA of microbes. Fun stuff, I promise you.